agenda, and then also all of you for being here. I know it is getting late, and it's been a really packed meeting so far. So thank you for making the time to come. Anyway, over the past day and a half, we have heard a lot of really inspiring talks that really focus on the technical side of free energy methods in many respects. How to make these methods faster, how to make them more accurate, and also how to increase their domain of applicability. I want to do something a little bit different in my talk. I would like to really focus on the practical application of FEP. And I would like to give everyone here a sense of what does it look like when we actually drive drug discovery programs using free energy perturbation. So the context, the story that I'll tell you that in is our SOS1 inhibitor program. This is a target that I'm sure is familiar to many of you. You may have worked on it. Since we're computational chemists and not biologists, I think it suffices to say that we want to develop inhibitors of SOS1 to block this complicated biochemical cascade. And this is a highly sought after target. Many other companies also have pursued this approach and have entered clinical trials with SOS1 inhibitors. We at Schrödinger in the therapeutics group launched our own SOS1 inhibitor program and culminated in the development of an IND-ready SOS1 inhibitor, SGR-4174. And today I'm going to tell you the story of how we used free energy perturbation to help achieve that goal. So one thing that we experience when we move into these crowded spaces is that there's a tendency for the chemical matter that inhibits these targets to look highly similar. And I'm showing you a little gallery here on the left-hand side that illustrates that. Most of these cores, as you can see, are highly similar to each other with this quinozoline type scaffold. And as Torsten pointed out to me earlier today, in fact, this is inherited from a kinase inhibitor previously. So the only exception perhaps is this phthalazine scaffold right here, but I think the point is pretty clear that these compounds look pretty similar to each other, including this northern piece here, which is a substituted phenyl ring. When we look at the crystal structures of these inhibitors in complex with SOS1, it's immediately obvious why this is the case. So the reason is that you need a heterocycle here to do pi stacking with this histidine 905. You want to form a hydrogen bond with this asparagine residue here. And typically you also want to fill out this hydrophobic pocket in the north with a T-shaped pi stack to this tyrosine. And that's really ubiquitous. That's the pharmacophore for these types of inhibitors. The question that we had and the goal that we set out for ourselves is to see if we could identify a novel and differentiated SOS1 inhibitor scaffold with drug-like properties. And the way that we wanted to do this was by seeking out new interactions in the binding site to leverage a novel scaffold generation. That, however, was easier said than done. When we did a really careful visual inspection of the binding site here, there did not seem to be too many interactions that we could repurpose or that we could go after that had not already been occupied by existing chemical matter. The only hint that there might be something different and new that we could do here was this triplet of acidic residues on the perimeter of the SOS1 binding site, GLU 902, 906, and 909. And what was very interesting when we ran an electrostatic surface calculation is we found that those three anionic residues actually form a contiguous region of negative charge, which we took to calling the solvent exposed acidic region. And that as well allowed us to formulate a hypothesis, which is that could we make salt bridges from our inhibitors to target these novel solvent exposed acidic residues as a potency handle for developing scaffolds? So before I answer that question, Dima asked me that I make this session interactive and we get some audience engagement. So I'd like to take a quick poll here. And if you think it's a good idea to target a solvent-exposed acidic residue with a basic group, shout out or raise your hand. I mean, who thinks this is a good idea? I see one hand, two hand, three hand, four hand. Okay, five hands. Well, that's about right. So, yeah, if you didn't raise your hand, you probably were more consistent with the thinking in the field, which is that targeting solvent-exposed acidic residues to form salt bridges is not a good idea. And this is probably one of the few topics that I have seen in the literature where every paper and review that we could find was unanimous in denouncing this idea. And we've excerpted 4 of these papers here. One of them, I think, suffices to read out loud. This is from a very famous article by Martin Stahl called A Medicinal Chemist's Guide to Molecular Interactions. And they say, we are not aware of any example in which charge-assisted hydrogen bonds on the surface of the binding site of a complex has led to significant affinity improvements. So that seems like a pretty clear non-starter. But again, in this discovery program, we didn't really have any other choices that we could see. And so we discussed it as a team. And the answer that the chemists came back to us was, well, look, we're willing to make the synthetic effort to go after this idea, even though it's unconventional, even though the chemistry is going to be hard. But given that this is going to be challenging, we would really like to know ahead of time that these molecules are going to be potent if we put the effort into making them. And that's really where we turn to free energy perturbation, or FEP plus in our case, to try to find these compounds and maximize the odds that even though they were pursuing this unusual idea, they would actually be worth the effort. So this is just a quick movie to show the thermodynamic cycle associated with FEP plus calculations. I'm sure everyone in the audience knows this already. But one thing that might be a little bit less appreciated is why FEP plus is so useful in these contexts. It's really not only about the accuracy. The accuracy is obviously necessary for pursuing these sorts of directions. And in this case, the accuracy that we've seen over quite a few programs is about 0.9 kcals per mole, which is 6 to 8-fold within experimental values. But it's also the speed and throughput that's really important. When we're running these discovery programs, we are on weekly DMTA cycles, and we really need to be able to triage a lot of compounds in a short period of time. And FEP plus lets us do that because it's fast, and that gives us throughput that enables the team to ideate very, very broadly. And over time, we build up these sorts of correlation plots where we get very good agreement between experimental and predicted free energies. So armed with FEP plus, we actually decided to test this hypothesis, even though it goes against the grain of everything that is in the literature, and I guess also the opinions of the vast majority of the audience here. So we started with this this scaffold right here, and we basically made it tricyclic. And what I mean by that is we added a third ring right here, and some of those rings were neutral and some of them were charged. The key point that I want to make is that all of these compounds were predicted ahead of time with FEP. And these compounds were made because of FEP. It's not correlative. It's not like the chemists had this idea anyway and we ran the calculation. They made these compounds in a causative way because the free energy calculations, in a somewhat surprising manner, predicted that if we put a nitrogen in this third ring that was ionizable, we would get a significant gain in potency relative to the parent scaffold and relative to putting a neutral third ring in the core. So having made these predictions, we actually went and synthesized and tested them, both for binding affinity and for biochemical activity. And indeed, what was really inspiring to see is that, as predicted, the neutral compounds were very weak, and the basic charged compounds were highly potent. And not only potent by a marginal gain in potency, but by significant gains in potency, to the point where these were about 750 to 1,000-fold boosts in potency simply by the introduction of a single heteroatom in these rings. So real, true activity cliffs that were prospectively identified ahead of time. To make sure that we were getting the right answer for the right reasons, we then subsequently got crystal structures of this tricyclic series. And indeed, what we found is that the basic group here, this nitrogen, is charged, and it recruits these two acidic residues, glutamate 906 and glutamate 909, to form a pair of salt bridges on the solvent-exposed perimeter of the binding site. So this was really exciting, and I think the whole team was inspired by this, and it broke open a new direction in the program, a new subseries that we wanted to pursue to see if it could help us achieve our goal of getting to an IND-ready candidate. So we played with this idea a little bit, and again, all of these compounds were FEP-driven, potencies and affinities predicted ahead of time. But the idea here was to see, okay, maybe we can take the nitrogen out of the ring and make it exocyclic. And we did that, and we made these azetidines and some of these homologated nitrogens. And in all cases, again, we predicted ahead of time that the charged compounds would be highly potent, both the R and the S enantiomers, isomers, and the FRET biochemical activity, which we subsequently determined backed us up. So this was, again, a way for us to gain confidence that what we had seen in that tricyclic scaffold was not a one-off, but this is a real phenomenon that we can control and that we can exploit to develop more drug-like chemical matter. I'm showing you on the right-hand side a representative snapshot of one of these compounds, compound 12, and although it's not a crystal structure, it is giving us the sense that indeed our hypothesis continues to be borne out, that recruitment of these two acidic residues, 906 and 909, is what's driving potency. Now, there's one slight complication here for those who are really experienced SAR gazers in the audience, which is that if it's true that the recruitment of these two acidic residues is what's driving affinity and potency, why is it that biochemically the potency of those two isomers is comparable, right? That doesn't really make a lot of sense because what I'm showing you here are the R isomers. And in the S isomer, right, the nitrogen is going to be flipped here up, which is pretty far away from 906 and 909. So I think this is another really great example of where FEP helped unravel this slight conundrum, because what we were able to see when we looked at the per residue interaction energies with glutamate 902, 906, and 909 is that the electrostatic, the interaction energy flips between the R and the S isomers. So for the R isomers, it's consistent with our mental model, which is that the gains in affinity are driven primarily by the interaction with 909 and 906 to some extent. But for the S isomer, the picture changes a little bit. Here, 906 and 909 are not as important, and it's really now the interaction with 902 over here that's driving the affinity gain. So I like this example a lot because it is something that was hard for us to rationalize. It's certainly not something that I think is obvious if you just look at these structures in PyMol, but it really illustrates how we can how FEP can weigh up this complicated situation, the long range, the short range electrostatics, to get to the right answer that's consistent with the experimental data. So at this point, we had a lot of confidence we could control affinity via this new interaction that we discovered, but that's not enough. We also have to have good ADME as well. And again, many of you know that a highly basic group is not fantastic for ADME properties. So we did the next natural thing, which was to prepare a set of homologated amines with withdrawing groups on the azetidine, fluoro, cyano, difluoro, the gem difluoro here, where we systematically drop the pKa of each of these compounds with stronger and stronger withdrawing pieces, from highly charged at 9.3 to neutral below at 5.6. Again, we predicted all of these affinities ahead of time, and I want to give a shout out to Ara, who mentioned the FEP groups tab yesterday. It's really important that we use that here because we need to include the contributions from both the charged and the neutral species to get these affinity predictions correct, which is what we did here. And indeed, as we subsequently synthesized and experimentally tested these molecules, we found that the potency dropped as we predicted it would. So the overall trend was that basicity and potency went hand in hand here. As the compounds got less basic, they also got less potent. Unfortunately, that presented a challenge because in these kinds of situations where we're trying to drop the basicity to improve ADME, we also were losing the potency that we really needed. And ultimately, even though we felt like we had a handle on what was going on here, we weren't able, in this tricyclic subseries, to solve these problems of high efflux, low oral bioavailability, some hERG activity, and just the fact that they were hard to make. So this is, again, where the teamwork comes in really front and center, and where the very tight integration we have between medicinal chemists and computational chemists is essential. And we took a step back and asked, okay, well, what can we do here to salvage this? And I also want to, because I think Dennis made a very important point in his drug hunter talk, which is that as we pursue these more complicated targets, also the molecules that we need to hit these targets are sometimes harder and a little bit more unusual. They're really what I call the off-diagonal compounds. And so we asked ourselves as a team, what can we do to find the off-diagonal compound and salvage that interaction? And so what we decided as a strategy was to preserve this interaction with the solvent-exposed acidic residues, this glutamate 902, 906, excuse me, 906, 909 with the basic group here. But to do something a little bit radical, we said, okay, let's just shave off the rest of the molecule. Let's just change course and see what happens, because now we know that this interaction is going to really anchor us in the binding site. Again, this is something easier said than done. As I showed you earlier, this binding is driven by a pi stacking interaction with histidine 905. And so we were, again, very assiduous about predicting all of these new cores ahead of time with FEP+. And it's not always obvious, just by visual inspection, why some of the cores were potent, as we predicted, and others were not. And just, I think this one example here suffices. This pyrimidone core was predicted to be 6.5 ppk in SPR. That was subsequently confirmed experimentally with no loss of ligand efficiency relative to the parent. But this pyridine here is inactive, just as we predicted it. So this is, again, an example of where we're threading this very small needle using FEP+ to make sure that we don't fall off the cliff. Now, this scaffold here was a really promising start because, as I mentioned, the ligand efficiency was comparable to the parent. But as I'm sure many of you noticed, we lost FRET activity because we had shaved off this R group here. Now, that R group was actually known to be essential for steric blockade, blocking the protein-protein interaction with KRAS. And so it seemed like a straightforward way to bring back the biochemical inhibition would simply be to add R groups here. And that's what we did with this library of spiros, some of these pyrazoles, morpholines, substituted cyclopropyls. And we were able to find in many cases that we could bring back FRET inhibition readily. And again, just to really be sure we were getting the right answer for the right reasons, we did get a crystal structure with this new core, this new subseries. And again, as we had predicted, the interaction with 906 and 909 is maintained. And here's the spiral group that's essential for the protein-protein inhibition. At this point, we felt as a team we were in a good place. We had this new core. We knew how to control it, and we felt really confident we could make accurate FEP+ predictions using it. So we did something that I think is a lot of fun. We opened this up to the broader team, and we basically performed an organization-wide scaffold hopping exercise where we used Live Design, which is our enterprise informatics platform, and let users from different teams sketch in ideas. The only prompt that we gave them was that we wanted their ideas to continue to target 906 and 909, and also to maintain that northern region. But other than that, we let them sketch in whatever they would like. because we were confident that we could predict a very wide range of new cores accurately at this point. I'm happy to say that this was very successful. It culminated in some very promising lead series where, looking across the board, the potency is excellent, the ADME is very good or acceptable in these cases, no risk, no DDI risk, safety is great, and the mouse IV, PK, and PO, oral and IV, looks good. Also, in a TGI model, these leads performed very well, and the PD was excellent. So to pull all this together, we were able to use this approach of repurposing this new solvent-exposed salt-bridge interaction that we had discovered to come up with these lead series that were potent, orally bioavailable, metabolically stable, and had low hERG risk. And ultimately, this culminated, as I mentioned, in the discovery of the IND-ready SOS1 inhibitor, SGR-4174. So this was really an exciting journey for the whole team. But at the end of it, we got a little bit curious, and we wanted to take a step back and ask, what's going on here? I mean, we really went in the face of a lot of convention. And is this just one specific N=1 type scenario? Or is there something a little bit more fundamental at work here? Have we discovered something that might be a little bit more general than just this one specific case? And it's actually really interesting because when you look in the PDB very carefully and cross-reference with SAR that's out there, this is not an isolated phenomenon. And there are two examples here that display a similar sort of idea of strong solvent-exposed salt bridges. One are these inhibitor series from Mirati. These are their KRAS G12D inhibitors. And they also have this really incredible matched pair here. We're going from a proton to this fused pyrrolidine system, gives you a 1,000-fold boost in potency, at least, via targeting glutamate 62, which is completely solvent-exposed. And then this interesting HPK reverse indazole series from Merck for their HPK1 inhibitors manifests something similar, where if we go from this piperidine to the morpholine to the piperazine, we get a 15-fold boost in potency from that single heteroatom. Again, but that's via targeting ASP101, which is solvent-exposed. So we did the final experiment here, which was just to make sure we could accurately repredict those match pairs with FEP. And in both cases, for KRAS G12D, we are able to get that tremendous gain in potency accurately predicted, as well as for the HPK1 series. And so I think for us, the big takeaway and the big learning from this experience was that we can use FEP Plus not only to drive our programs forward, but also to learn something a little bit more general, a little bit fundamental, and maybe something that was just underutilized or not fully appreciated in the field. So anyway, if you are working on any programs that have solvent-exposed acidic residues in them, give it a shot. And maybe next year at the Free Energy Workshop, we'll hear some more exciting stories of where this has panned out. Anyway, thank you again to everyone for taking the time to listen to me. Naturally, this was the work of a large group of people. I also want to thank in particular the leadership, Robert, for providing us with the extraordinary computational resources that we enjoy at Schrödinger, and our CRO partners as well. The work is published, so if you're interested in what you heard, feel free to have a look in more detail, and happy to take questions now, and feel free to email me or grab me at the poster session again later. Thanks.
Thanks, Abba. I think we have time for maybe 2 questions. We have a question over here.
Those are all great questions. So we didn't predict these with ABFEP. The reason is that we had a congeneric series. And I think those follow-ups that you suggested are really interesting ones that we could dig into. I have not yet, but I think it's a great future direction. And I think you're putting your finger on what really needs to be done here, which is, why does this work? Like, what is going on here? What is the mechanism that explains why this is the case? So I think if this is the group of people that will figure this out, and yeah, I think those would be really good future directions to look into.
There you go.
Yeah. Yeah, no, it's a good question. I think this is a good example of where the interaction diagram really provided a clearer picture of what was going on than just these specific discrete interactions. But both are useful. I mean, the two pictures were consistent with each other, but it was very clear when we looked at the per residue energies what was happening. I think that's the kind of thinking. And also there's significant water interactions here as well.
Right. Let's thank Abba again.
Thanks.